Hello dear viewers, welcome to Crown Biology classes. This is Rohit speaking, and in today's lecture I will be discussing the giant chromosomes, that is the Polytene and the Lampbrush chromosomes. So let's start. Some cells at certain particular stages contain large nuclei with giant or large sized chromosomes. These are different from chromosomes in their structure because these are of large size. The giant chromosomes are the Polytene and Lampbrush chromosomes. So first of all, discussing the first giant chromosome, that is Polytene chromosome. It is also known as salivary gland chromosomes. An Italian cytologist E.G. Balbiani discovered them. E.G. Balbiani in 1881 had observed peculiar structures in the nuclei of certain secretory cells, that is, of salivary glands of Chironomus. Those peculiar structures were nothing but salivary gland chromosomes or the Polytene chromosome. The structures were long and sausage shaped and marked by swellings and cross striations. Cross striations were the transverse bands. The structures that are the Polytene chromosomes. Unfortunately, E.G. Balbiani did not recognize them as chromosomes, and his report remained buried in the literature. But in 1933, Theophilus Painter, Ernst Hitz, and H. Bohr rediscovered them in Drosophila, and they recognized them as the chromosomes. So, for the very first time in 1933, by Theophilus Painter, Ernst Hitz, and H. Bohr recognized them as the chromosomes. The present name, that is the Polytene chromosomes, was suggested by Kohler due to the occurrence of many chromonemata in them, as numerous chromonemata were present in them, and that's why Kohler named them as Polytene chromosomes. This is the diagram of Polytene chromosome of an insect showing bands and interbands and a puff or Balbiani ring. Chromosome puff is also known as Balbiani ring. As you can see in this diagram, the chromosome puffs are diffuse swellings along the regularly banded arms of Polytene chromosomes. Dark band and interband can also be seen. These are the transverse bands present between two dark bands. Interband is present. Chromonemata is nothing but a coiled chromatin thread within a chromosome. It is a coiled mass of threads. Some cells of the larva of the dipteran insects such as flies, example drosophila, mosquitoes and midges, Chironomus, become very large having high DNA content, as these polytene chromosomes are formed when the products of multiple rounds of S phase, that is the synthesis phase, remain tightly associated to form one supersized chromosome. This point is very important. Polyteny of giant chromosomes is achieved by replication of the chromosomal DNA several times. There will be multiple rounds of S phase without nuclear division, and the process is termed as endomitosis. In the process of endomitosis, the nuclear envelope does not rupture, and as a result of that, no nuclear division takes place and no spindle formation takes place. In fact, polyteny differs from polyploidy. In polyploidy, what happens is there is also an excess DNA per nucleus. In both polyteny and polyploidy, excess DNA per nucleus will be present. But in polyploidy, the new chromosomes are separated from each other, but in polyteny, the chromosomes do not separate. The alignment of many chromomeres gives polytene chromosomes their characteristic morphology, as there will be many chromomeres are present and these chromomeres are arranged in which a series of dark transverse bands alternates with clear zones called interbands. This we have already seen in the diagram, that there occurs transverse bands, there occurs dark bands, and in between two dark bands there lies interbands. The clear zones are called the interbands. About 85% of the DNA in polytene chromosome is in bands and 15% is in interbands. See, very less amount is present in interbands, but in bands 85% of the DNA is present. Chromosome puffs or Balbiani rings. Chromosome puffs or Barbiani rings, these are the swellings of bands of the Polytene chromosomes, and in which what happens where the DNA unfolds into open loops. When the DNA unfolds into open loops, and that is as a result of intense gene transcription. These Balbiani rings are exceptionally large puffs on the Polytene chromosomes, and these play a role in RNA and protein synthesis. Bierman and Baher in 1954 have studied the fine structure of these chromosome puffs or Balbiani rings. As you can see the diagram of Polytene chromosome, the first one is the line drawing of Polytene chromosome of Drosophila melanogaster female. You can see various arms of the different chromosomes, interband region and bands can also be visible in between two dark bands. Interband is visible, and the diagram of Balbiani ring or the puff is magnified. Chromonemata is visible, interband and dark band are also visible in this, and Balbiani ring is also visible. The second type of giant chromosome is the Lampbrush chromosomes. The Lampbrush chromosomes were first observed in salamander, that is an amphibian oocytes in 1882. So for the very first time, Lampbrush chromosome were discovered or observed in salamander, that is an amphibian oocytes in 1882. The Lampbrush chromosomes, they look like the brushes which were used for cleaning the glass chimneys of old fashioned paraffin or kerosene lamps, and hence they got their name Lampbrush chromosomes, and they were first described in detail in shark oocytes by R. Ruckert in 1892. And where does this Lampbrush chromosome occur? The Lampbrush chromosomes occurs at the diplotin stage of meiotic prophase in the primary oocytes of all animal species. So this position of these Lampbrush chromosome is very important. Diplotin stage of meiotic prophase in the primary oocytes, and both vertebrates and invertebrates possess these Lampbrush chromosomes. These are also found in spermatocytes of several species, giant nucleus of acetobacteria, and even in plants. And these are best visualized in salamander oocytes because they have a high DNA content. And for the very first time also these were discovered in or observed in salamander oocytes. Since the Lampbrush chromosomes are found in the prolonged diplotin stage of meiotic prophase 1, they are present in the form of bivalents, in which the maternal and paternal chromosomes, these are held together by chiasmata, and these chiasmata are the sites where crossing over has occurred. So these are the sites for the crossing over. The paired homologues are not condensed as usual chromosomes would be. Those paired homologues are bivalents. These are not condensed. Instead, they are very long and stretched out. Instead of condensation, they will be stretched out. Each bivalent has 4 chromatids, 2 in each homologue, and the axis of each homologue consists of a row of granules or chromomeres from which lateral loops extend. These all points will be clear when we see the diagram in the upcoming slide. The loops are always symmetrical, each chromosome having 2 of them, one for each chromatid. The loops can be categorized by size, thickness, and other morphological characteristics. So this is the diagram beautifully explaining the arrangement of Lampbrush chromosomes. As you can see, maternal and paternal chromosomes are present, and there are sites where these maternal and paternal chromosomes meet, and those sites are known as chiasmata, and chiasmata are the sites of crossing over. In the enlarged section of a chromosome you can see the bivalents stretched out, and they are forming the chromatin loop. Chromomere is also visible. And in the last diagram, what is visible? Chromatin loop is visible, chromatid is visible, and chromomere is visible. And each loop appears at a constant position in the chromosome, and this fact helps in chromosome mapping. As you can see in the diagram, A is the enlarged view of a part of Lampbrush chromosome. There is main axis of chromosome present and loops are also visible. These are the stretched out bivalent chromatids. Matrix is also present which consists of protein as well as RNA. And B is the diagram of one loop of a Lampbrush chromosome in which chromosome axis is present, chromomere pair is visible, matrix which contains proteins as well as RNA, and filament which is only made up of DNA is visible. Ending this video on a positive note, there is a saying which says, "Don't stop until you are proud." Thank you for watching.